right. So there we go. That's your title slide. So today we're talking mainly about receptor tyrosine kinases and a little bit about ligand-gated ion channels, although we kind of did that with synaptic signaling, so I don't spend very long on that. And we're talking about the uses for these receptor tyrosine kinase pathways. Okay, so the part of the protein, we're going to draw that together to the right, that sticks out of the cell is going to function as a receptor. So what you're going to see, why their name sounds so weird, is because it's a molecule with two ends. The end that sticks out of the cell is a receptor. The end that faces the cytoplasm is a tyrosine kinase enzyme. It's kind of like a split personality. Okay, so what this says in there, in case you can't see that, is TYR for tyrosine kinase. So this is going to be your receptor end. It's going to be facing the outside of the cell. So the part of the protein that extends into the cytoplasm functions as a tyrosine kinase enzyme. And so remember what we said yesterday about kinases? And so we said kinases are kind of like Robin Hood. They steal from the rich and give to the poor, except in this case, they are stealing from a molecule that is fairly rich in phosphate groups like ATP, and they are giving it to another molecule. So the name, the word that comes before kinase is usually what's receiving the phosphate group, okay? So the fact that this end of the molecule is a tyrosine kinase enzyme, the kinase part, as you'll see today, will remove phosphate groups from ATP, but they're going to add phosphate groups to these tyrosine molecules that are facing the cytoplasm. Okay, so let's get into the pathway. That's really the only background information that you need. We'll be doing a little drawing. This pathway just isn't as long as the one from yesterday. So how does a receptor tyrosine kinase work? Well, first, a signaling molecule has to bind to the receptor end. Okay, and I know I had to draw this a little small, so my membrane's going to have to be teeny tiny. I didn't really plan that out super well, but that's all right. Because that part's got to be in the cytoplasm, that part's got to be out of the cell. And so there's my phospholipid bilayer. All right, and these right now are two separate receptor tyrosine kinase proteins. And so this is going to be the signaling molecule, like insulin, that would bind to them. Okay, so underneath that, the binding of the signaling molecule to the receptor tyrosine kinase causes two receptor tyrosine kinases to get very close together, forming something called a dimer. So to start off with, those two receptor tyrosine kinases are separate. But when a signaling molecule such as insulin binds to each one, it's going to cause them to get closer together and basically become attached to each other. And that's something called a dimer. A dimer has kind of like repeating subunits. It has subunits that are like all the same, or it's made of 2 or 3 or 4 of the same kind of molecule. So a dimer is a chemical compound composed of 2 identical or similar subunits or monomers. So there's my insulin that bound to that receptor. There's my insulin that bound to that receptor. This thing that I have made here is a dimer. And so I'm not going to draw in a ton of phospholipids, but I'll just draw in a few so that you can see where those are located. Okay. All right, so this thing that we have just formed, this is called a dimer. All right, next. Okay, so the formation of the dimer activates the tyrosine kinase enzyme part of the protein. That's the part that's facing the cytoplasm. So they are still going to be a dimer. So they still look like this. Phospholipids are here. Signaling molecule in this case is insulin, so there's your insulin. And I'm gonna put TYR for tyrosine in there. I know it's kind of tiny, but there's basically 6 tyrosine kinases on the part facing the cytoplasm. So yesterday, when we were doing that pathway, how did we show that the G protein was activated? Yeah, we did like the yellow around it. So we're going to do the same thing here today. If we're saying that the formation of a dimer activates the tyrosine kinase end of the protein, then we need to draw our yellow halo around that part. Okay, once activated, the tyrosine kinase part of the protein catalyzes the transfer of a phosphate group from ATP to the tyrosines on that molecule. So because you have 6 tyrosine kinases, and the kinase part is going to be what's taking the phosphate group off of the ATP and adding it to the tyrosine, this is going to require 6 molecules of ATP because you're taking 1 phosphate group off of each of them and adding it to where our glowing tyrosines are now. I just have to put a T because I had to make the picture smaller. All right. We gotta show that they're active. Because this is the tyrosine kinase end. So if we have an ATP molecule, it's gonna have 3 phosphates. So we have adenosine, which is the adenine nitrogenous base plus the ribose sugar. And then we're gonna have 3 phosphate groups. We're going to have 3 phosphate groups. And we know that the energy in this molecule is stored in the bonds between the phosphate groups. So now that this tyrosine kinase end of the protein is active, it's going to do its job, which means the kinase part, again, remember Robin Hood, it steals from the rich, it gives to the poor. It is going to steal the phosphate group off of here, and it's going to add it there. So if I showed this 5 more times, I'm not saying you have to, I'm just saying if you need that visual, phosphate, phosphate, okay, this kinase enzyme takes that phosphate and it's gonna add it to each tyrosine. So when we call it a tyrosine kinase, the kinase is removing the phosphate groups from the ATP, but the tyrosine part tells you what protein or amino acid you are adding the phosphate groups to. So I'm not going to write all those ATPs, but I am going to put a phosphate group on each one. So this would have cost me 6 ATPs to do this step. So far, so good. Okay, let's keep going. So the now phosphorylated tyrosines attract relay proteins, which are just other proteins that are going to do a specific job from inside the cell. So I can make this kind of big. Okay, so the formation of the dimer, we said, activates this tyrosine kinase end of the molecule that is facing the cytoplasm or projecting into the cytoplasm. And so here I'm just showing you where your cell membrane would be in relationship to all this. The receptor end has got to stick out of the cell, while the tyrosine kinase end is going to face the cytoplasm. So, in the last step, we said that kinases steal phosphate groups from ATP and transfer them to the tyrosine molecules, and we've got 6 of those. So that will take us 6 ATPs to do that, and I'll make 6 ADPs by the time I'm done. So we got to add in our phosphate groups. And then we said these phosphorylated tyrosines are going to attract relay proteins, which are just other proteins in the cytoplasm that can help carry out a specific function in the cell. They're going to want to come and bind. That's a relay protein. These are all relay proteins. They're going to come bind to where the phosphate group is. Okay. So remember how yesterday, how we said that when we had the activated G protein, and that activated G protein bound to adenylyl cyclase, that adenylyl cyclase became active as well. And then when the G protein acted as a GTPase enzyme and converted the GTP to GDP, that now the G protein was inactive and it's separated from the adenylyl cyclase, so the adenylyl cyclase was inactive. Well, because these relay proteins are attached to these activated tyrosines and phosphates, that's going to cause these relay proteins to become activated as well. Because a lot of times in the cell, you can have inactive versions of various proteins, like we saw during the phosphorylation cascade of our pathway yesterday. We had inactive protein kinase A. Cyclic AMP activated it. We had inactive phosphorylase kinase. Okay, protein kinase A activated that. And so that's kind of what's going on here is right now these relay proteins are inactive, but they will become active here. So now we have those phosphate groups on the tyrosines, and we said that is going to attract those relay proteins. But then this says once the relay proteins bind to the phosphorylated tyrosine, that the relay proteins change shape, and this activates them. So I'm going to change it to a heart shape. Okay. So now we change their shape, and that activates them. How are we going to show these relay proteins are activated? We're going to do the yellow halo around it. Okay, and these activated relay proteins then typically are going to unbind, and they are going to go trigger a cellular response, which can be a lot of different things. So in the case of insulin, the cellular response is going to be the insertion of glucose transport proteins into the cell membrane so that glucose can be taken into the cell. Because remember, that's kind of what insulin does. Insulin is kind of like putting the key in the door to unlock the door. The door would be like the glucose channel. So when insulin is present, glucose can come out of the bloodstream and into the cells. All right, so much shorter pathway. I know it's a little vague to say cellular response. It was a little more gratifying yesterday with the G protein pathway to know, okay, I know this is what's going to happen. I know my endpoint is going to be the production of 20,000 molecules of glucose, which are going to serve as fuel for my muscle cells when I'm in an emergency. Here, we just say that those relay proteins are ones that will go off in the cell and, cause various things to happen. It just depends on what kind of cell we're talking about. So then we're going to do some application of this pathway. And yeah, not much. Notes aren't too terribly long today. So one of the things I would expect you to be able to do is look at a diagram like this, pick out what's familiar. Okay, I would ask you to, I could show you a diagram like this. It involves growth factor and a growth factor receptor, and I could say, what kind of pathway is this? Is this a G protein pathway, or is it a receptor tyrosine kinase pathway? How I'm going to tell the difference is I'm going to look at the receptor. So see here how we have two receptors that are joined together to form that dimer. And then they have a cytoplasmic component to that receptor. Did the receptor protein we talked about yesterday, did they have that? No, they didn't form dimers. They didn't have a cytoplasmic component. So if I showed you this pathway, even though you're not familiar with the whole thing, you should be able to look at that receptor and tell me that this is a receptor tyrosine kinase pathway. Okay? And so this is activating, this phosphorylation cascade is activating a series of different proteins in the cell. Notice, now they do this a lot on the AP exam. Well, they'll give you a big diagram of a pathway you are unfamiliar with, but they're expecting you to be able to kind of decipher a little bit about what's happening. Okay, so here, whatever this MNK1 is, this protein is going to make it into its active form. And then we see translation, the production of additional proteins, cell growth, and division. Okay, that's like mitosis. So notice what it says at the bottom. Promising target for cell-specific anti-cancer drugs. Because cancer happens when your cells divide too often. Okay, so then we have whatever's going on down here. Some of the proteins are going to go bind to your DNA and cause the transcription of genes promoting cell growth and division in your nucleus. Okay, so a couple things. Yeah, you should be able to kind of look at a diagram and pick out some things that are familiar to you, and you should definitely be able to look at that receptor and tell that that is more similar to a receptor tyrosine kinase than it is to a G protein linked receptor. So this is a specific example. And if her ears are ringing, today is actually her birthday. So my cousin Tracy was diagnosed with non-small cell lung cancer in 2016. Her only symptoms were a persistent cough that she couldn't get rid of. And you know, a lot of people are getting sick right now. And so a lot of us have a cough or we have congestion or that. That's really all she had. It was just this cough that wouldn't go away. When the doctors did a chest x-ray, they found a grapefruit-sized tumor in her right lung. Tracy was 51 at the time and barely smoked in her late teens and early 20s. So we've kind of got to get over that stigma of, oh, if they have lung cancer, they must have smoked. Because a lot of people who get lung cancer didn't smoke or didn't smoke enough to cause it. And so this is a type of cancer that typically affects non-smokers who are on the younger end of the spectrum. So the little bit she might have smoked in her late teens, early 20s did not contribute to this. This cancer affects non-smokers. So in the summer of 2016, doctors attempted to remove the tumor, but when they saw how it had infiltrated the surrounding lung tissue, in other words, tumors kind of have these extensions. Okay, that's where cancer kind of got its name, because you know how like the zodiac sign, like Cancer is the crab, and it's got these legs that kind of stick out. That's what cancer tends to look like under the microscope is, yes, there are too many cells there, but it's kind of got these extensions that are a lot of times wrapping around other tissues. So they ended up they made the incision, they went and looked at it, and they're like, oh. And so they ended up having to remove her entire right lung. So you can live with just one lung. She just has to be super careful, especially in the age of COVID and the COVID that's coming back, whatever version this is now. Doctors then did genetic testing on her to see if she might possibly have a cell signaling defect. Now I messed up the formatting. Doctors then did genetic testing on her to see if she might possibly have a cell signaling defect associated with the cancer, which she did. So this is not for me to like tell a big giant story about my family, but this is the direction that cancer treatment's moving into. The fact that she had this genetic defect means she has to take a pill every day for the rest of her life that will block her receptors. But it's basically blocking those cells from dividing. So this cancer shouldn't come back. And she never had to do traditional chemo, which just targets any rapidly dividing cell in your body. And that's what causes you to lose your hair and be really, really tired and all that. So how are tyrosine kinases involved? What's the connection? So it turns out that she had an overactive type of tyrosine kinase receptor called anaplastic lymphoma kinase. You don't have to know that for your test. The medicine that they prescribed for her was called Xalkori or crizotinib. And then this is kind of where the thinking comes into play. We look at this next slide, we want to see what looks familiar. And we want to see what we can figure out from these pictures. So I see the individual tyrosine kinase. I see it here where something's bound to it, so it formed a dimer. Okay? It says down here, how does crizotinib seem to work? And I drew you a little picture of where it affects this pathway. So you know enough now to be able to answer that question. What does crizotinib do? It's blocking something. What's it blocking? Blocking what happening to the ATP?
Phosphates attracted to different types.
Yeah, that, you know how we said the tyrosine kinase, how kinase likes to steal from the rich and give to the poor. And by rich, I mean ATP. It's rich in phosphates. It has 3 phosphates. So it is blocking that tyrosine kinase end of the protein from being able to remove phosphate groups from ATP and add it to themselves, which means the relay proteins inside the cells will never get activated. And that is basically stopping these cells from dividing. Okay, because all this other stuff happens. And when it says proliferation increase, proliferation, I'm spreading. It means I'm making more and more and more cells. So you should be able to do something like that. Like if I gave you a diagram and I said, okay, what process in this pathway is this interfering with? You should be able to tell me that from a diagram. Okay. All right. This next slide is important because they always seem to ask about it either in the multiple choice or in a free response. And it's really kind of common sense, but we need to take a close look at this. All right, let's look up top first. Because I see it either on the free response or the multiple choice every year. All right. So in this one, this one, this one, and this one, we have the same signaling molecule. That's the little red triangle. In the first 3, we also have the same receptor. Okay, but in this first pathway, I'm going to activate this relay protein, then that relay protein, and I'm going to get this response. Here, this first protein can activate either of two different relay proteins, so I can technically get two responses, okay? Here, again, I'm activating a different protein, so I'm getting a different response. And that's how I can get 4 different responses using the same signaling molecule and the same receptor. It's because my relay proteins were different, okay? That's what that is trying to show you. And then we'll look at the right-hand side of the diagram in just a second. It says common in tyrosine kinase receptor pathways, since different internal proteins, meaning relay proteins, can be activated. Here, we have a different signaling molecule binding to a different receptor, so we know that the effect of that is going to be different. Here we have the same signaling molecule binding to a different receptor, and also there's different relay proteins as well. So I forget how they word these questions, but I think they show you a bunch of pictures and say, which one of these would result in a different response than the original or something like that. So you've got to realize that all those greenish turquoise color shapes there represent the relay proteins. Okay, now on this next slide, I'm trying to think why I put this in there, but I think I put this in there because maybe I was doing a little flashback to yesterday. The sympathetic division of your nervous system controls your fight or flight response. And fight or flight has to do with epinephrine. And so these are the effects that epinephrine has on your different organs. So it dilates your pupils. It lets more light in so that you can really see what's going on. It speeds up your heart rate. It dilates your bronchi. That's why people who have allergies, pretty severe allergies, carry an EpiPen around with them so that if they eat something that maybe they're deathly allergic to, they can inject themselves with that EpiPen, that epinephrine, and its natural response is to dilate your airways. And that will keep your airways open until they can get you to the hospital and give you more treatment. So if you're ever around somebody, because didn't this happen, so last year or the year before with one of our track guys down at TL Hanna? Do you remember that? Okay. And so he ate something or drank something, but he was allergic to something in there, right? And so if you're ever in that situation where you've got to help somebody out and he says, just put the EpiPen in me, you still have to get him to a hospital. EpiPen doesn't mean, oh, everything's fine now, because they need to be checked out further. But that is why it's called an EpiPen. It's epinephrine. It dilates your airways so that he can still breathe while you get him to the hospital. But the reason I put this up there, I believe, was this conversion of glycogen to glucose. So remember that was the endpoint in your G protein pathway. Okay, flip to the next page. We're just going to talk briefly about ion channel receptors, but we kind of already did that with synaptic signaling. And then I'm not even going to go over that last slide. All right. Erase that. Okay, so a signaling molecule ligand binds to a receptor in the membrane. This causes the gate part of the membrane, the channel part, to open. So this would be like a ligand-gated ion channel. So we talked about this with acetylcholine, how acetylcholine binds to its receptor, and that opened ligand-gated sodium channels, and sodium would rush into the cell. Once the ligand unbinds from the receptor, the gate closes, and that particular ion can no longer pass through. And then don't have to worry about that last slide. But what I do have to show you is the Kardashian kinase analogy for this pathway. So you guys may think this is silly, and it probably is. But for those of you who need a little help memorizing this, it's all good. This will work. Because I'm not going to ask you to write out these steps. I'm just really asking you to put them in order, both on your quiz tomorrow and on your test on Thursday. Okay, and I'll put this up on the big screen as well. Okay, so this is your Kardashian kinase analogy. All right, now I know Bruce Jenner doesn't really look like that anymore, and he is now Caitlyn, and I respect that. But it probably works a little better with my pathway if he's still Bruce here, so we're going to think like way back when they first met. Okay, so who's this? You don't know her name? That's Kris Jenner, right? And so Kris Jenner, and when he was Bruce Jenner, before he became Caitlyn, like I said, this is back when they first met, so he was still Bruce then. And so basically Kris and Bruce meet, and they get married, and that forms the dimer, okay? So them getting married forms our dimer. And then luckily we have 6 Kardashian-Jenner children. So we've got Kim and Courtney and Khloe, Kylie, Kendall, and Rob. Okay, so that kind of goes along. Each of them is a tyrosine kinase enzyme. And so the formation of the marriage results in, even though not technically, these 6 kids. Those 4 were already around. Those 2 came out of the marriage. You with me so far? All right, then remember, tyrosine kinase catalyzes the transfer of 6 phosphate groups from 6 ATPs to 6 tyrosines on the molecule. So, in our little Kardashian analogy here, fame and fortune are going to be our phosphate groups. Because let's face it, Kris was kind of like their manager and orchestrated a lot of them becoming famous. And so we're going to represent the tyrosine kinase stealing a phosphate group off of 6 ATPs. That phosphate group is going to be fame and fortune. Okay? So the formation of the dimer activates them. So if the parents weren't married, maybe they would have never existed in that case or became famous. And then now that they're active, they seek fame and fortune, each in their own ways. Fair? Okay. So now that these Tyrosines are phosphorylated. What should happen next? What does that attract? Not that it's another R word. Those relay proteins, right, from inside the cell. So guess who the relay proteins are going to be? That's going to be their significant others. And I don't know how outdated this is, so forgive me. It's probably really outdated on your sheet.
Travis
Yeah.
is with her.
Right? He's not with her anymore, is he?
Just a beard. Who is that?
I don't
Bottom left. Devin Booker is the middle.
You know who all these people are?
No. Just the.
I mean, I only know who that is.
Is that Pete Davis?
That's Pete Davidson. They're not together anymore. So they're going to be the relay proteins. Now, some of these people, yes, were already famous in their own right. Some of them, like think about, well, who was Courtney first married to? What's his name? He's kind of a dirtbag.
Y'all supposed to know this.
I don't know. Why are we supposed to know this?
You know what?
His name is Scott Disick. Okay, and she has children by him, but he's kind of the definition of what happens next, which is now that these tyrosines are phosphorylated, and remember the phosphorylation is the fame and fortune, that's going to attract these relay proteins. And so some of them, I'm sure it's pure fairy tale love. Some of it, like with Scott Disick, I think was a little opportunistic because he wanted to have fame and fortune too. So them being phosphorylated drew in these relay proteins. And now through association, okay, Pete Davidson was famous kind of in his own right, but definitely got him up a level when he started dating her. So by them associated with them, that is now going to activate these relay proteins, and then they are now famous and have fame and fortune and can go off and do their own thing. So even once they break up, once these relay proteins detach, they can go off and do their own thing. Good? It's not perfect, but for some of you, that's how you're going to learn this pathway. And you don't have to say who you are, but some of you, that'll really help you learn this pathway.
What did you just say?
All right. Just give me a second to figure out what I want to do next. I'm going to end my recording, and then we'll figure out what we're going to do because I have another lab set up in lab.